Hello, good afternoon, everyone. We are going to be starting lecture 8 today, which is heredity. And we're going to learn about the basis of how we produce sperm and egg, which is called meiosis, and that's related to mitosis. There are many similarities, but of course, there are some differences as well. So we're going to spend the first part of the course learning about first part of the lesson learning about that, and then we're gonna switch to solving genetics problems. Class average for quiz number 3 was pretty high. It's 75%, so not bad at all. We only have one more quiz coming up for the course, and that's not happening until like 2 weeks later. So I'll tell you more about it when more people join. So meiosis, right? The purpose of meiosis is to produce gametes, right? So gametes are basically sperm or egg, right? So I think it's coming up, but let's write it down first. Gamete equals to sperm in male or egg. And these are, of course, required for sexual reproduction. Previously, when we learned about mitosis, we understand that some organisms use mitosis as a way for reproduction, but that would be called asexual reproduction, where there's only one individual and they make copies of themselves. But in sexual reproduction, which is what humans and many other animals do, there is going to be the fusion, the combination of reproductive cells from two parents. The father supplies the sperm and the mother supplies the egg, and the two come together to form an offspring. So by now, you should know we have 46 chromosomes in all the cells, except for the gametes, except for the sperm and the egg. Sperm has 23 chromosomes and eggs have 23 chromosomes. And so when they come together, 23 plus 23 gives you back the 46 that is needed to create a full set. And so the process to produce these gametes, the process to produce the sperm and egg, that is called meiosis. Now, humans are diploid organisms. Diploid means we have two full sets of chromosomes. And again, we know this already. 1 to 23 from mom, that's one set. 1 to 23 from dad, that's two sets. So collectively, two sets of chromosomes, we say we're diploid. The gametes, the sperm and the egg, only has one set of chromosomes. So we call them haploid. In this picture, we can see the adults, 2n equals to 46, and n here, n equals to the number of chromosomes per set. So two sets would be 2 times 23, which is 46. And when we undergo meiosis in the ovaries and in the testicles, that creates the haploid gametes, right? Only one set. And when fertilization occurs, when the two things come back together, then we're going to recreate the diploid cells. Okay, so haploid and diploid, all the cells that diploid in the body, except for the sperm and egg. Now, in other organisms, it's possible to have more than two sets. If it has 3 sets, then we say triploid. 4 sets, then we have tetraploid. But generally, we can just call them like, if there are more than 2, we can say it's polyploid. Polyploidy, in fact. Right, so strawberry has 8 sets. This is yam. Some people call it sweet potatoes. That has 6 sets. Right? The plant tends to have more sets than animals, but it's not uncommon in some animals either. Right? Some animals like sometimes can have more than 2 sets. 2 is not like the norm. That's I guess that's the point I'm trying to make. Right? Humans have 2 sets, but it's entirely possible to have more than that in other organisms. So one of the questions that I was wondering, growing up is how do they make seedless watermelon? Right? The seed is needed to grow a plant, of course, but if the seedless watermelon has no seed, how do you grow more seedless watermelon, right? So it turns out to do that, there are regular watermelons, which are diploid, 2 sets of chromosomes, and then they have a special breeding technique that causes the watermelon to become tetraploid. So now we have 2 kinds of watermelon. We have the watermelon that has 2 sets, and then we have watermelon that has 4 sets. So if you take the seeds from the 2N, right, the seed, it will be haploid, right, just like our sperm and egg. And what about if you take the seed from this one? What would the seed be for the 4N, from the 4N watermelon? What's the N number for the seed?
2N.
2N, that's right. So if you combine these together, right, if you fertilize them together, that actually 2 plus 1 becomes 3N. And the point here is anything with odd number set of chromosomes are sterile. Okay, you cannot have odd set number of chromosomes and be able to undergo sexual reproduction. I should rephrase this. Anything with odd number sets of chromosomes cannot undergo sexual reproduction. Okay. Remember, sexual reproduction means you have to create the gametes. Humans, animals is sperm and egg. In plants, it's like pollen, right? But when you produce the gametes, you have to divide the genetic material by half. So if you have an odd set of chromosome, like over here, like 3N, you can't divide this by 2. Like what's like 3N divided by 2? You cannot have like 1.5 of a set, right? So anytime you have odd sets of chromosomes, you cannot do sexual reproduction because you cannot create gametes. You can still do mitosis, that's fine, but meiosis is not possible. So that's how you get the seedless watermelon. Because the seedless watermelon, they cannot produce seed. The seeds are like the gametes, right? The seeds are like the sperm and the egg, so to speak, right, of the plant. So that's how it's possible. All right. So somewhere out there, there are the 2N and the 4N watermelon, and if you breed them together, you get the 3N. Okay. So where does sexual reproduction, where does the production of gamete occurs? It turns out the process of sexual reproduction begins with meiosis in germ cells that are found in the gonads. So that's a lot of words here. Okay, so first of all, germ cells, like people get confused. Sometimes they hear germs, they think of like bacteria and stuff like that. But germ cells are cells that will become sperm or egg, depending on which sex you're talking about. And you will only find germ cells in the gonads. The gonads are reproductive organs. So testis for male and ovary for female. So in the reproductive organs, in the gonads, that's where you will have the germ cells. And the germ cells are diploid themselves, 2 sets, but after meiosis, they would become haploid. Okay. Now, a zygote is what's formed when a sperm fertilizes an egg. Right? So like in this picture, you can see there are many sperms trying to gain entry to the egg, but only one will make it, right? Like you and I are the product of that one sperm getting into the egg, right? Yeah, so like millions of sperms are trying to compete, but only one will make it. And as soon as DNA is injected into the egg kind of forms like a barrier to prevent other sperm from injecting the DNA. And the end result of fertilization is a zygote. So the zygote will undergo rapid divisions, and eventually it becomes the embryo, right, which is the baby. So here is meiosis overview. We started with 2N, and then after meiosis 1, we have 1 division. And then meiosis 2, we have a second division. Okay. Now, it's a little bit confusing, but remember how we count the chromosomes. If they're stuck together, we count it as 1. So for simplicity here, we only see 2 chromosomes here. 2 chromosomes. Okay, 2 sets. If that's 1 set, that's another set. And over here, after you copy them, after you do DNA replication, it is still 2 sets. But now the chromosome has a copy of itself, right? It has a sister chromatids. And then you separate them. So you have 1 set, and then you separate again, and then you have 1 set. Now that's a little bit confusing. I totally understand. So what we're gonna do is we're gonna draw it out, and hopefully after drawing it out, it's gonna make more sense. Okay, so get some paper. You're gonna need 2 colors, at least 2 colors. Probably a highlighter is best. Okay, highlighter is probably best because it's easier to color with them. Okay, but here we go. This is class notes, March 25th, 182. There we go. Oops. So we're gonna do meiosis. There we go. Now stop me if I'm not making sense, okay? I haven't done this in a bit of time, so I'm a little bit rusty with the drawing. But hopefully it'll make sense. Now, again, how many chromosomes do we have? Just checking. Anybody? How many chromosomes do we have?
46.
Yeah, 46. But I'm not gonna draw 46, but it's confusing, right? So I'm gonna draw like maybe like chromosome 1 here from mom, and then chromosome 1 from dad. Sorry, chromosome 1 from mom. Chromosome 1 is chromosome 2 from mom. Chromosome 1 from dad. And then chromosome 2 from dad, okay? So that's 1, 1, 2, 2. And of course, there are 3, 3, 4, 4, all the way to XX or XY, right? So recall, there are some terms that I have to refresh your memory on. And this is chromosome 1 from mom, chromosome 1 from dad, right? 1 and 1. We call these homologous. Homologous chromosomes. Okay, they are same length, same size, and they have same order of genetic information. Okay, but the genetic information might be different, right? The alleles might be different, blue eyes, brown eyes, but the order of genetic information is the same. If it's hair color, eye color, blood type, then it's same thing. Hair color, eye color, blood type on the other one. You're gonna find it at the same location, same locus.
So, Vincent, the length of the chromosome person to person is different?
No, it's the same. For everybody, it's the same. But what I mean is like 1 and 1 are the same length, and then 2 and 2 are the same length, and then it gets shorter and shorter as we go on. But you know, 4 is same length as 4, so on and so forth. So your chromosome 4 is same as my chromosome 4 in terms of length. But it will be shorter than 1, 2, 3 and longer than, you know, 5, 6, 7, 8, so on and so forth. Does that answer your question? Okay. So here, this is meiosis. So let's take a look at what's going to happen in the process of meiosis. So at the end of interphase, you would have duplicated all the genetic material, and so what I want to focus on is prophase. Prophase 1. Okay, so meiosis is separated into meiosis 1 and meiosis 2, and inside meiosis 1, you would have prophase 1, metaphase 1, anaphase 1, and telophase 1. Same thing for meiosis 2. You would have prophase 2, metaphase 2, anaphase 2, telophase 2. So let's focus on prophase number 1. Okay, prophase number 1. What happens in prophase number 1 is that, you know, the chromosomes have duplicated already. Okay, chromosomes would have duplicated already, but they would find each other. They would find each other. So homologous chromosome find each other and form a tetrad. So a tetrad is like 2 X's, if you think about it. So that's like chromosome 1, 1, 1, 1, 2, 2, 2, 2. This whole thing is a tetrad. Now, this does not happen in mitosis. In mitosis, don't draw this, but in mitosis, after duplication, they just like randomly exist in the cell. The pairs don't find each other. But in prophase 1 of meiosis, they find each other. So that's a difference between mitosis and meiosis. Do you guys understand what I mean here? So how many tetrad are we gonna have in prophase 1? How many of these, you know, pairs are we gonna have? How many tetrads?
2.
I mean, if you consider all the chromosomes in a human cell, in a regular human cells, that I did not draw.
46. 10.
Okay, I hear 46. Anyone else?
23.
23. Yeah, 23, right? Okay, because look, this is already chromosome 1 from mom and dad. Right, so they're together already, and then 2, 2, 2, 2, 3, 3, 3, 3, all the way just up to the sex chromosome, right? So there will be 23 of these tetrads. So the purpose, what is the purpose of forming the tetrad? Homologous chromosome can exchange genetic information by a process called crossing over. Crossing over. So I'm going to show you what this looks like. Crossing over. So a little bit of the blue will switch with the red. A little bit of the father chromosome is going to switch with the mother chromosome, so to speak. So like that. Do you see that? So this over here, right? This is like crossing over. It allows the exchange of genetic information between the paternal and the maternal chromosome. And that creates genetic variation. Now, I am only showing you a little bit of exchange here and there. In reality, the exchange is going to occur throughout the entire length of the chromosome. Okay, so it's more accurately, it would be like, you know, red, blue, red, blue, red, blue alternating, because it keeps on switching, right? But it's very difficult to draw that, and it's very confusing. So I'm only, you know, showing a little bit of the exchange at the bottom here, right? Do you guys understand that?
Vincent, this happens in gonads. This just happens
That this happens
in the
when we
gonad.
produce sperm and eggs. That's right. Do you guys understand this situation here?
No, no.
No? Okay. Is it my drawing or is it my explanation? Okay.
Like just like go over it one more time.
Sure. Okay. Don't draw this, just watch. Okay. Tetrad, right? You understand this is tetrad? Yeah. So
Yes.
what happens is this. Okay? This part here, I'll try my best here, okay? This part here breaks off, and this part breaks off like that. And then they're like, okay, let's switch. There we go. Okay. So in the end, your mother chromosome, it's now gonna have a little bit of like father chromosome on it,
Oh,
right? And then same thing for the father one's gonna have a bit of mother one, right?
okay. Thank you.
Better, yeah. So listen, right? There is no point exchanging genetic material from here and here, right? Because those are sister chromatids. They're sister chromatids. They are identical. So if you switch them, like even if you switch them afterwards, is, you know, doesn't do anything, right? So I'm just gonna explain that to you right here. The crossing over only happens between non-sister chromatids. Okay, and it creates genetic variation. Okay, so like how come your kids inherited the father's eyes and not the mother's eyes? Well, maybe the mother chromosome switched the eye gene with the father chromosome, and then that get passed on, right? So depending, so like there's this big shuffling of all the traits between the mother and the father when we're producing the sperm and the egg. That's why our kids don't look identical to us, right? Make sense? So that's prophase number one. All right, moving on. We're gonna go to what's after prophase? You can just think about it in your head. We're gonna go to
Metaphase.
yeah, exactly. Metaphase 1, right? Of meiosis. So metaphase 1, yeah, sorry, go ahead.
Can you up a little bit?
Can I what a little bit? I'm
Yeah,
sorry.
drag up. Yeah, drag up.
Oh, up a little bit. Okay.
Yes. So in here in prophase 1,
Yeah.
we call it tetradias when
Yes.
they cross each other. So still in the second one, when they cross, still their name is tetrad or
This whole thing?
yes.
Yeah, that's still a tetrad. That's right. Yeah,
Okay.
and the crossing over is referring to the part where they, you know, literally cross over and do the switch, right? And do the exchange.
Okay. Yeah.
Yeah. So in metaphase, don't draw this, but listen. Like in mitosis, metaphase, they all line up like this, right? You remember that? This is mitosis, right? Now in meiosis, is different. In meiosis, instead of individual chromosomes lining up in the middle, we have the tetrad lining up. And we have the tetrad lining up in the middle. Let me just draw that nicely. There we go. We have the tetrad lining up in the middle. And so there's the tetrad. And then there is the other tetrad. And I want you to understand something here. I want you to understand that how the tetrad lines up is a random process. In the first tetrad here, in the first tetrad over here, we can see that the father copy is on top and the mother's copy at the bottom. And then the next one, we have the mother copy on top and the father copy at the bottom. But that's completely random. It could be like father, mother, and then the next one is also father, mother. Right? So the orientation is random, and that adds to genetic diversity. Now I'm just gonna put the centriole there to help you keep track of where things are. That's the centriole. These are the spindle fibers. Right, so here, tetrad lines up in the middle, as opposed to individual chromosome, okay? And more importantly, the orientation of the tetrad, of each tetrad, is random. Further contributing to genetic variation of the gamete. Is that okay so far? Any follow-up questions? Okay, watch the videos that I post in the video resource. When you see this in animation, it probably will make a lot more sense. But anything so far? Okay, after telophase, after metaphase, we have anaphase. Anaphase 1. Right? So the spindle fiber is gonna pull on the tetrad, and the homologous chromosome will separate. The homologous chromosome will separate. And so this is the centriole again, centriole, and it's going to be pulling the things over. And here, we're going to have the father copy up, and with a little bit of the mother one because of the crossing over that happened earlier. And over here. Okay. And over here on the second pair, we have like this. And then we have this. Okay. So the homologous chromosome gets separated. And finally, we have the telophase 1. So we have 2 cells. We have 2 cells right here. Okay, so the top cell, we're gonna have this thing with a bit of red, like that. Now the other one. Like that. Right? So of course this is gonna split, right? But look, we started with, hold on a second, we started with 2 sets of chromosome. Right? 1 set, 2 set. This is 2n. Sorry, this is 2n right here, 2n. And after the first round of division, right, look at how many set of chromosome we have now. Just 1 chromosome, 2 chromosome. So this is n now. That's the end of telophase 1. That's the end of meiosis 1, essentially. Okay. Now I'm not gonna go through the whole meiosis 2. Meiosis 2 is exactly same as mitosis. There's not much difference. But I want you to see this. Okay, see if you understand this. Okay, so this is meiosis 2. And then we're gonna have cells right here. Okay. So each of these are gonna get separate, right? Again, in anaphase 2. And the cells might have, focus on this for now, okay? Look at it. You're only gonna get half of this thing, right? Because they separate during anaphase. So like a cell might get like this one, or it might get the one with a bit of the mother chromosome. Same thing for this one. You only gonna get half of it because again it separates in anaphase 2. So you might get the one that has a bit of the father one, and then this one is just that one. So that's also n and n, right? Now focus on this one here. The gamete might get the one with just the red or the one with a bit of blue. And same thing for this. You either get just the blue or the blue with a bit of the red. And it's all like randomized, right? And these are all n as well. So in the end, you get 4 genetically different haploid cells. Okay, 4 genetically different haploid cells. Questions? Okay, I want to go to the study guide and I want to fill in this table with you. This is question number 3 in the study guide. All right. Can you please go get that? Hold on a second. I think the one I posted online is more, I updated something. Let me just double check here. Yes, I've added like an anaphase to it. So give me a second. Let me just update this thing. There we go. Just delete that one. Okay, so question 3. Let's fill this in together and see if it makes any sense to you. Help me out. Show me that you understand what I'm trying to show you. How many cell divisions do we have in mitosis? How many times does the cell split in mitosis? Anybody? Mitosis, the stuff that was on your quiz. How many times does the cell split?
One.
Yeah, one time. Right? One time. Once. Split. That's it. Right? Original cell, and then you get 2 cells. Number of daughter cells produced? 2. Meiosis. How many divisions? How many times does this split?
2.
2 times. Twice. Okay. Probably should draw it sideways this time. And then each of these will split again. So how many daughter cells?
4.
Yeah, 4. But I'm gonna put a star here. Okay? And I'm gonna just write a quick note. 4 for men, and then 1 for female, for women. Okay. I'll explain later. How about the daughter cells? Are the daughter cells diploid or haploid? Diploid means full set, haploid means 1 set. For mitosis?
Diploid.
Diploid, yeah. And haploid for meiosis. We're talking about humans here. So the number of chromosomes in daughter cells are 46. And only 23 in female. Sorry, in meiosis. Okay, prophase. Yes.
Sorry, is that because of tetroid?
Because of a tetra?
Tetroid?
No,
Yeah,
not because of a tetra. Because it's split twice, right? It's split twice.
Oh, yeah.
Yeah. Okay, so in prophase of mitosis, the homologous chromosomes don't find each other, right? I was telling you. So like you have chromosome 1 here, chromosome 2, and then you have another chromosome 1 here. Let's just draw it like randomly here, and then chromosome 2 here. They do not find each other. Right? Homologous, sorry, it's not enough space. Homologous chromosome do not find each other. Okay? They're not going to find each other. They're just going to be randomly scattered in the cytoplasm. But as we learn, for meiosis, they will find each other. They will find each other. Why do they want to find each other? Tell me. What's the purpose of finding each other?
To exchange genes?
That's right. So homologous chromosomes form tetrad to undergo crossing over. It creates genetic variation. I keep on saying this, but it's true, so that our kids don't look identical to us. It's a blessing for them and for us, right? So that is the difference between prophase and prophase 1. It looks much better when you put them side by side. Okay. Metaphase. Metaphase, right here. So in metaphase, the chromosome lines up in the middle, right? Like that. Right? Individual chromosome. Lines up. In the middle. Okay? If you have space, you can put in the middle. I don't have space. But for meiosis, what lines up in the middle?
Tetrads.
Tetrads.
Yeah, they're tetrad. Very good. Tetrads line up. In the middle. I have space for in the middle this time. Okay, so big X, you know, like that. Now, of course, the crossing over would have happened already. The red's gonna have a bit of blue and the blue is gonna have a bit of red. But that's not the point of this picture, right? This picture is just like a side-by-side comparison, right? So I'm not gonna draw it. You can draw it if you want. You can put a bit of blue on it and so on and so forth. Okay. Anaphase one. Anaphase one. In anaphase one, I guess I should put the centriole there as well. You know, with the spindle fibers, centrioles, spindle fibers. Now in anaphase, sister chromatids separate. But in meiosis, it's not the sister chromatid that separates. It's the homologous chromosome that gets separated. And again, there's going to be crossing over that has already happened, but I'm not showing it for simplicity. So I hope this side-by-side comparison makes it more, you know, understandable and easier to comprehend, huh? That's all. Okay, genetic relationship between the daughter cells and the parent cell, identical. But for meiosis, it's different. What kind of cells undergo mitosis? Somatic cells, bodily cells. And then here is germ cell, right? The ones in the gonads. That's going to become the sperm and the egg. Function to grow and develop. Repair damage. Okay. And that's it for human. We're talking about humans, right? For meiosis is formation of gametes. Do we have any questions before we move on? Okay. So this is just the picture with some words. You can read it through. It's pretty much what I taught you, right? If you scroll through them, you can kind of see them, you know, separates like that. And then you get your gametes here. We just compare mitosis and meiosis, so that's done. Now let's talk more specifically about what happens in male and female. So meiosis, since we are producing gametes, it's also called gametogenesis. Genesis is the creation, right? Creation. So gametogenesis is the creation of new of gametes, of sperm and egg. In male, it takes place in the testis, and or testes for plural. Females, it happens in the ovaries. Male is called spermatogenesis, creating sperm. And for female, it's called oogenesis. Because the egg is called oocyte. Okay, so you know, oogenesis. Sperm production is pretty standard. It's basically meiosis, like what I told you. But for oogenesis is a bit more complicated. So I'm going to show you the difference. Start with the spermatogenesis. We have in the germ cell. This is the germ cell. It's called a spermatocyte, and it's going to be in the testis. And this is meiosis 1. So after the first division, it's already 23 and 23. But remember, they are stuck together. We just count them as one, right? And then second round of division, you separate the sister chromatid this time around. And then there are going to be 4 sperms, and each only having 23. Now in female, it's, you know, similar, pretty much the same thing. But when it's going through the telophase, when you separate the cytoplasm, it's called cytokinesis, right? You might remember that. The division of the cytoplasm is not 50-50. The egg is very big, right, compared to the sperm. And so when you create the egg, you don't have the luxury of dividing the cytoplasm into 50-50. Instead of having 2, you know, medium-sized egg, what your body does is it gives most of the cytoplasm to one cell, and then the other one just gets very little. So like 90%, 10% split. So the one that has very little cytoplasm, we call that a polar body. The polar body might or might not divide further, but regardless, the polar body is not going to become the egg. The polar body is just going to be reabsorbed by the body, and it's not useful. So this cell that gets most of the cytoplasm was going to undergo another round of division, and the same thing happens. One cell gets a lot of cytoplasm, the other does not. So we end up having just one egg. Okay. So 4 genetically different sperm from each cell that undergoes meiosis in male, and 1 egg for female. So spermatogenesis occurs in the testes, starts with diploid spermatocytes and generate 4 genetically different sperms. It's a continuous process. It produces, I mean, like after puberty, right? Of course, produces 200 to 300 million sperms daily. That is a lot, right? That's almost 2,000 to 300 per second. But of course, only half of them becomes viable. Okay, so there's like a QC process, a quality control process, and only viable ones are gonna make it out. So that's spermatogenesis. For oogenesis, it's you know, a little bit complicated. Happens in the ovaries, but in female, the person is actually born with all the eggs that they're gonna have in their entire life. Okay, so it's a discontinuous process. Before birth, the oocytes already began the process of meiosis, but they stop in prophase 1. Right? So there's the tetra, there's a crossing over and stuff like that, but it suspends in that state until puberty. Right? So when puberty comes, once a month, prophase 1 will move to metaphase 2 during ovulation. So only 1 egg is chosen, right, so to speak, for each cycle. Sometimes there are more than 1 eggs, and that's how people get like non-identical twins. And then it gets suspended in metaphase 2. The process is only completed after fertilization. Okay. So that's why the quality of the egg tends to degrade over the course of a person's life. people with decided to have kids later, their eggs are more prone to have been damaged throughout life, and the risk of getting chromosomal disorder increases with the age of gestation, of becoming pregnant. I mean, so one of the error that could happen is what we call like non-disjunction. Non-disjunction is a situation that occurs when chromosome fails to separate during anaphase 1 or anaphase 2. And they're supposed to separate, right? But if they don't separate, then it's called non-disjunction. It's much easier if I show you a picture than to read this to you. So over here, let's look at the situation on the left first. Okay, so for simplicity, we're just tracking 2 chromosomes. So let's say we separate it in meiosis 1, so no problem. They separate, the homologous chromosome separates, right? But then there is a non-disjunction that happens in meiosis 2. Okay, so on this side here, that's normal, but there is a non-disjunction right here. Okay, that's what the arrow shows. So instead of separating these 2 sister chromatids, one cell gets all of it. So when the sperm fertilizes this egg, then there is going to be an extra chromosome. Whereas here is going to have missing chromosome. Right? These ones are okay though, okay, because both the separation were fine. Now let's go back to here. It says non-disjunction in meiosis 2. One of the final gametes will have additional chromosome, and one of the final gametes will be missing chromosome. That's what this is, extra and missing. Okay. If, say, the chromosome that's extra is chromosome 21, then the person will have 3 chromosome 21. And we call that trisomy 21. Does anybody know what condition that is? If a person is born with 3 chromosome 21?
Down syndrome?
That's right, Down syndrome. But it's not just chromosome 21. There are other chromosomal disorders. Trisomy 18. There's many of them. Missing a chromosome, on the other hand, is usually lethal, though. Okay, if the baby is missing 1 chromosome or missing part of a chromosome, usually that's not gonna grow to full term. Okay, what if non-disjunction happens in meiosis 1? So let's take a look. These things are supposed to separate, but they didn't. So one cell gets all of it, and the other cell is missing. So the missing one will continue to miss that pair all the way through, while the one that got the extra will continue to have extra all the way through. So here, both of these have extra, and then both of these will have missing chromosome. So it's much worse to have non-disjunction earlier on than later on. Okay. If non-disjunction happens in meiosis 1, all the eggs are going to be problematic. But if it happens in meiosis 2, then you know, only half of it will be affected. There's still a chance that you're gonna have a good normal egg. So
Vincent,
mhm.
so in the previous slide, when this happens, according to the eugenesis chapter, meiosis 2 starts after fertilization. Yes,
Yeah.
yes. So what happens with the spermatogenesis part? Does it not have any, like any
Impact?
Yeah.
No, so spermatogenesis is an ongoing process. I mean, non-disjunction can happen as well in spermatogenesis. But here's the thing, right? Like the men produce like so many sperm every day. The
Okay.
chances of having a sperm that has non-disjunction that actually makes it out of the body and then actually goes to the egg and fertilize it, the chance of that is much lower than if the mutation happens in the egg, right? Because the egg, you know, it's precious. There's only one comes out, right? You know what I mean?
Oh, okay.
Yeah.
Okay. Thank you.
So
I have a question too.
sure, yeah.
So when, for example, can you back the slide? Yeah, in here. So, for example, if in one child happen like Down syndrome, so it's only from the women's side, means, for example, is not related to sperm.
Theoretically, the sperm could cause the same problem as well. Okay, but as I just mentioned, it's unlikely because there's like so many like non-disjunction is not a common occurrences, right? So even if it does occur, it's like I'm just making this up. Okay, let's say 2 sperms out of millions of sperms, and what's the chance that those 2 sperms is gonna actually fertilize the egg, right? It's pretty low, right?
Yes.
Okay. But then for the egg, because there's only one egg, so if that egg comes out and then it gets fertilized, then the problem is gonna arise. And the problem is like the chances of these chromosomal disorder does increase. I think I have a graph, maybe it's in the second semester, but there's a graph that shows the incidence of chromosomal disorder and the age at which a woman has a child. So if they have it later on in life, the chance of the egg having some kind of chromosomal problem also increases. And that's why people like freeze their egg, right? Like celebrities, they do that, right? If they decided to have a kid later on, then the egg is still kind of fresh, right? Yeah, I don't know if does this answer your question or you have a
Yes,
follow-up.
exactly. Thank you.
Yeah. So of course, nobody wants this to happen. It's a terrible thing, and so there are techniques to screen for these kind of things. And what a karyotype is, a karyotype is an orderly arrangement of chromosomes. So you can see here the chromosomes are lined up. And when I say they line up, I don't mean like physically line them up under the microscope. Long story short, you basically take a picture of all the chromosomes and then you line them up digitally, like on a computer screen. Okay, and then you have them all laid out here, and that can be useful for screening for genetic disorder. So, for example, we can see that this is a male, and all the chromosomes look fine. There are no extra chromosome or missing chromosome or anything like that, so that's fine. Over here, this one, there's an extra chromosome 21, right? So that's trisomy 21, and you can do this before the child is born. There are 2 main methods of doing this. One is called amniocentesis. For those of you who don't know, okay, the baby grows in the womb, and then the womb, as in like the uterus of the woman, right, which is right here, and the baby is surrounded by these fluid called amniotic fluid. Okay, you might have heard that when you before you give birth, you wait for the water to break, right? Okay, so the water that comes out when it breaks, that's the amniotic fluid. So that fluid, keeps the baby floating inside the mother, so it's not as heavy for the mother, and it protects the baby for quite a bit. And that fluid contains cells that are kind of washed off from the baby. So what you can do, I mean, what the doctor can do is they can use ultrasound as a guiding tool. They could insert a needle through the belly button, I believe, and they could extract a little bit of the amniotic fluid. And then they could take the cells from the amniotic fluid and basically look at the DNA, look at the chromosomes. Okay, so that can be done 15 to 20 weeks of pregnancy, and the parents would be able to be informed about what's going on, and make plans and decisions accordingly.
As you said, this chromosome will be seen like this in a prophase stage. Okay, so when they extract the fluid, they will let the cells to divide and to see
Oh, I see what
these
you mean. Yeah, no, I mean you don't have to look at the cells under prophase or whatever. Is that the cells? All cells have chromosomes, right? So if the cells have extra chromosomes, then all the cells in the body is gonna have the extra chromosome, right? So you don't necessarily have to be looking at like cells that are actively dividing. Okay.
But in that phase, we see like chromatin, not chromosome, right?
Oh, I see what you mean. Yeah, yeah, yeah. So at any given time, they actually do what's called like a metaphase spread. At any given time, there are going to be cells that are undergoing some kind of division, right?
Oh, yes.
Yeah, because there's so many cells in the body, right? At any instant, there has to be a percentage of the cells that are dividing, right? So it's not a difficult find. Yeah, but good question. That's very good question.
I have a
Another,
question.
sorry, yeah, go ahead.
Yeah, in amniocentesis, so when the woman like 15 or 20 weeks,
Hmm.
the doctors know in the X-ray, for example, as we know, every here is a lot of problem happen. For example, I saw so many people, but if the treatment, the amniocentesis, uh, the doctor, how they identify the amniocentesis happen in one mother,
Okay,
or
yeah. Or what? Sorry, or finish your question.
until the baby born.
Mm. Yeah.
After that,
Okay.
how they know this amniocentesis.
Right. Okay. So first of all, it's not X-ray, right? It's ultrasound that they do.
Okay,
But
ultrasound.
yeah,
Yeah.
I understand your point. Yeah. So amniocentesis is not a treatment. Okay. Amniocentesis is not a therapy for anything. Okay.
Mm,
Amniocentesis is just a technique
mm.
to get a bit of the baby cells and then examine the under the microscope. Okay, extract the DNA, extract the chromosome, and look at it under the microscope, right? So, if I'm understanding you correctly, you were saying like, what would prompt a doctor to perform amniocentesis? Like,
Yeah.
yeah. So, like, there are conditions where it runs in the family, as you will learn later on in the lesson, right? So if the mother is high age, for example, if there is a family history of a particular kind of genetic disorders that can be picked up by a karyotype, not everything can be picked up by karyotype, okay? Some things you just can't, but some things like Down syndrome, something like extra or missing chromosomes, those you can pick up by karyotype. So if there is a if the doctor suspect that there's a
Okay.
higher risk of these kids having inherited these conditions, then that would be an option for the mother to do, right? Maybe sometimes they do a scan, okay? They do a ultrasound and they see something abnormal on the scan. Uh, then they could suggest a follow-up amniocentesis.
Okay. Okay. Thank you.
Yeah. So the other method is called CVS, chorionic villus sampling. Okay, chorionic villus sampling. So the chorionic villi is they're these tiny projections that are coming out of the placenta tissue. They look like fingers, literally like that. Okay, and that is the same genetic material as the fetus, as the baby. So you can take a little bit of a sample of those projections coming off from the placenta, and then you can extract the cells from it and examine it. This one is done through the vagina, and you can do it a little bit earlier in pregnancy, okay, 9 to 14 weeks. So, I mean, it's a sensitive topic, but then for some people, abortion might be like an option, right, for them. And depending on where you live, beyond certain weeks, that's not going to be legal or safe or possible for some women. So CVS is like a biggest advantage is that you can do it like way earlier compared to amniocentesis. Okay, but both of them has the same goal, is to get a little bit of the cells from the baby, indirectly, of course, and then you look at the chromosome. Any other questions before we move on? Okay. After meiosis 1, the cell is considered as, now this is pretty straightforward, so I'm just gonna do it for you and you can check it out later. Okay, haploid. Which of the following is true regarding human gametes? Let's do this one. Let me start the poll for you. Go ahead. Okay, thank you. The answer is they are all haploid. Okay, gametes are all haploid. They are produced in the testes and the ovaries. Ovaries, not ovida. Okay, a little bit of female anatomy for some of you. These are the ovaries. They're the fallopian tube or the oviducts that connects it to the uterus. Okay, so the oviduct is where fertilization occurs. That's where the sperm is gonna meet the eggs if it's gonna happen at all. Okay, this one is complicated. Let's do it together. Which of the following is true for an organism that has a chromosome number of 2n equals to 6? Okay, some of you, I mean, all of you should know how to solve this, right? If you solve for n, what's the n number?
8.
8. Yeah, 16 divided by 2 is 8, right? So that means one set of chromosome has 8 chromosomes in it. The n number is the number of chromosomes per set, right? Number of chromosomes per set. Okay, per set. So let's see. The organism has 16 sets of chromosomes. No, I just told you is 8. A gamete from this organism has 4 chromosomes. Okay, so look, it has 16 chromosomes in total. 16 divided by 2 is 8. The organism is sterile. No, it's only sterile if, earlier I told you about a watermelon, only sterile if it has odd number sets of chromosome, which it does not. So it's okay. Each cell has 8 homologous pair. Well, it has 8 chromosomes per set. So that's chromosome 1, 1, 2, 2, 3, 3, 4, 4, all the way to 8, 8, right? So that would be 8 homologous pair. That's right. Okay. The organism is diploid with 16 chromosomes per cell. That's all. Okay. I think I haven't been giving you breaks for like many lessons now. We can have one now. I won't make it for Wednesday, but I will post a recording of a lecture. Okay, so you guys can watch that. I'll post it at the lecture link. So you don't come for a synchronous class on Wednesday. You just watch the recording, and then we'll resume next week. Okay, feel free to send me questions as well. If you don't have trouble with the recordings, if you don't understand things like that, but it's a pretty straightforward lesson. Okay, you should be able to watch and learn from that. But let's take a quick short break. Okay, we'll resume at 1:45. See you in a bit. All right, let's keep going here. I have some good news for you guys. For quiz number 4, I mean, it's usually people struggle a little bit because there's like more problem solving happening in there. And just to give you a little bit of an advantage, you will have 2 attempts for quiz number 4. So you can do it once, and then you will see the mark right away. And if you want to do it again, you can do it again. Right? The questions might be different, but you get to do it twice, and only the best mark will be recorded. So hopefully that's gonna help you guys a little bit. Yeah. Moving on. Now we're going to talk about the molecular basis of heredity. And this guy here, his name was Gregor Mendel. And he is often called the father of genetics because he was credited with discovering how traits are passed on from one generation to the next in peat plants. And that kind of formed the basis of modern genetics. So let's go through some terminologies first. A gene, you should know by now, is a segment of DNA on a chromosome that encodes for a protein. And the genes might have different versions, which we call alleles. Now, if we are diploid, we have two of each chromosome. And if the genes are the same on those chromosomes, then we say homozygous or pure. On the other hand, if they are different, then we say heterozygous or hybrid. So here is an example. We have 2 chromosomes, one chromosome from mom and one chromosome from dad. And the alphabets here are the different genes. So for gene A, you have capital A for one and you have small a for the other. They are not the same. There are 2 different versions of the gene. So for gene A, the person is heterozygous. You guys learned about homogeneous and heterogeneous solution, right? In chemistry, I assume. Yes? No? Maybe?
Yes,
Yeah. Homozygous means homogeneous means same, right?
same,
So,
yes.
yeah. So homozygous also means the same. So A is heterozygous, B is heterozygous. Do you guys find anything that's homozygous here? Which gene is homozygous or pure?
The second one?
D.
D, that's right. D is homozygous because it's small d and small d, right? E is also homozygous. Anything else? Maybe one more?
I, L, M.
Yeah, exactly,
H.
right. I and L and H, right? As if they're the same allele, then we say homozygous or pure. If they're different, then we say heterozygous or hybrid. Pretty straightforward.
Vincent, the order of genes in all human are the same. Like,
Yes.
for example, first of all, I color. Yeah. Okay. Thank you.
Yeah. So, like for example, where are you gonna find the gene that codes for insulin? It's gonna be on chromosome 7, position whatever, like the page number, right? The locus, right? It's gonna be the same for everybody, which is important later on, okay? Because once we find out where our gene is, as you will learn in next week, we'll be able to clone it. We'll be able to mass produce that gene, okay? And that's important in biotechnology. So a character is a heritable feature, such as like flower color. For human, give me an example of a heritable feature in human. Anybody?
A skin color.
Yeah, skin color is heritable. Anything else?
Hair type.
Yes, yeah, curly hair, straight hair, right? Yeah, very good. Another one, maybe? Just one more.
Height.
Yeah, to a certain extent, yeah. Tall people tend to have kids that are also tall. Okay, so 2 things here. Number one, genes is not everything. Okay, we talk about the concept of penetrance, right? Okay, just because you have a gene doesn't mean you're necessarily going to express it. So there is something called nature, as in what kind of genes you inherit, that you don't have a choice. Nature versus nurture. Okay, nurture. So nurture, that's the environment that you grew up in. Right? So even identical twins, they don't grow up to be looking identical, right? Like there are subtle differences, and in some cases, like big differences between how they look. If you have twins that grow up in different continents, for example, they would grow up to be quite different actually. So the environment has is a big factor on how the genes are being manifested. Okay, so not everything is nature. Some things are nurture. My point is that height is yes, there's a genetic component to it, but it also has to do with growing up, right? What kind of environment, do you get nutritious food and that kind of stuff. So that's a character, okay? And a trait would be a variant of the character. So purple flower, white flower, okay, darker skin, lighter skin in humans, and things that we acquired are not heritable. Okay, so like if I work out a lot and I become like super muscular and strong, my kid is not going to be born super muscular necessarily, right? Or for that matter, they're not necessarily going to grow up super muscular, because I work hard to train to get those muscles. Okay, so that's not going to get passed on. That's something that I have acquired throughout my life. Like an easier example to understand it, like if you dye your hair purple, your kid's not going to grow up to have purple hair, right? Okay, unless they too go dye it, right? So not everything can be passed on, right? Those that can pass on, those that has a hereditary component to it, then we call it a character. Now, some trait are dominant trait, others are recessive. If it's dominant, it will always show up. Right? Even if you have one copy of it only, it will show up. And we use capital letter to represent these things. I'll give you some examples to clarify very soon, okay? So if you don't 100% get it, don't worry, it's coming. Recessive means it will only be expressed if you do not have the dominant allele. And we use small letter to represent these things. So here are a bunch of examples of what's dominant and recessive in people. Okay, so if you are able to roll your tongue like that, that's a dominant trait. If you cannot, then it's a recessive trait. Freckles, you can't really see it here. Freckles are like those little, spots, right, that people might have on their cheek, cheek, face. That's dominant. No freckle is recessive. This kind of hairline is called widow peak. That's dominant. Straight hairline is recessive. And then your earlobe and cleft in the chin, whether you can bend your finger back or not, right? Those are dominant trait. 5 fingers is recessive. 6 fingers, that's dominant. Do you know some people have 6 fingers? Okay. So, what's the deal with this? Well, the genetic makeup of an organism, that's called the genotype, whereas the phenotype is going to be the outcome, the expression of the gene. So let's just use a very simple example. Let's pick one thing here. Okay, rolling the tongue. That's dominant. So we use capital letter for it, right? So big R equals to roller. You're able to roll your tongue. Small r means non-roller. Okay. So we have 2 chromosomes, which means you can obtain 2 alleles. So if a person is big R, this is homozygous or pure, right? Homozygous or pure. Does this person roll the tongue or not roll the tongue? It's not a
Roll
trick question. Yeah, this is roller, right? Okay, no problem. Roll the tongue. Small r, non-roller. I cannot roll my tongue, so I am a non-roller. What about someone who is big R, small r? Can they roll the tongue? Sorry, Mary, what is it?
Roll the tongue.
Roll the tongue, that's right. Because the dominant gene is there. The dominant allele is there, right? If there's a capital letter, that's gonna show up. So two people can have different genotype. Their genetic makeup are different, but their phenotype are the same. You understand? Let's pick another thing here. Let's pick I don't know, free hanging and attached. Like you can look at your earlobe, right? Like it's dangling or is it attached, right? Mine is dangling. I don't know about yours. Mine is free hanging. So free hanging is big F, and then attached is small f, okay? Small f. So what if we have two people who have attached ears? Are they big F or small f? Attached ear.
Small f, small.
Yeah. So let's say the father is small f, the mother is small f. What does it mean? The whole family, all the kids are gonna be small f, right? All the kids are gonna have attached ears, right? Because why? The father is small f, so the only thing he can pass on is a small f. The mother is also small f, so the only thing she can pass on is a small f. And then together, the whole family is going to be small f. Do you understand? Any questions? Okay. What if the parents have free hanging ear? Both of them have free hanging ears, like the earlobe. Is the whole family going to have free hanging ears? What do you think?
Yes.
No.
Yes? No? Okay. Anyone else? You don't have to answer. Like, think about it. Right? Well,
Yes.
yeah, okay. Well, let's see. If the whole family is free hanging, what's the genetic makeup of the father? Let's say the father is free hanging. What is he? Is he big F, small f? What is it?
Big F. Two big Fs.
Big F, right? But actually, it can be big F, small f as well, right? Both is possible. So if the father is big F, small f, and the mother is also big F, small f, then there is a chance that the kids are gonna have attached ear as opposed to free hanging. Because the father can pass on the small f and the mother can pass on the small f, and the kid's gonna have small f, right? So we're gonna go through a few more of these examples to help you understand. So here we go. Mendel's did his experiment with pea plants because they're easy to breed, and plants you can fertilize them very easily with just a paintbrush. You probably do that if you grow tomatoes indoor. You can use like a Q-tip to fertilize them, so and they grow pretty quickly. So that's why he used a pea plants. And here are a bunch of traits that he figured out. So dominant seed is round, and then the recessive one is wrinkle. Yellow is dominant, green is recessive, so on and so forth. So he has two laws. And the first law is that it's called the law of segregation. During the formation of gametes, each gene separates from each other so that each gamete carries only one allele for each gene. Well, that makes sense because during meiosis, the chromosome separates. So even though you are big F, small f, so like on one chromosome, it's big F, on the other chromosome, same chromosome, homologous chromosome is small f, you're gonna separate these chromosomes. Right? Half of the gametes is gonna get the small f, the other half is gonna get the big F. All right? You're not gonna get both. You're only gonna get both if there is a non-disjunction, right? Which doesn't happen under normal circumstances. So that's what the law of segregation means. Okay, the chromosome separates. I mean, he didn't understand, he didn't know about meiosis at the time, but he observed that the tray separates. So we're gonna do what's called like monohybrid cross. Monohybrid cross means we only look at one tray. Okay, we only study one tray at a time. So P is the parent generation, and he started with pure round seed. So that's big R, is specified it's pure, then we know it's homozygous, right? And pure wrinkle seed, so that's little r. So let's take a look at the first generation, the F1 generation, offspring of the parents. So in this situation, let's take a look at the gametes produced. Gametes is like the pollens, right? Produced by this particular seed. Well, this seed is big R. So when the chromosomes separate, half of the gametes are gonna carry the big R, and so does the other half, because that's the only option, isn't it? As for the wrinkle seed, it's small r and small r. So, you know, when they separate, half of the gametes get small r, the other half also gets small r. So if this gamete fertilizes that gamete together, the offspring is going to be big R, small r. Similarly, if this gamete fertilizes with that gamete, it's going to be big R, small r. Right? This and this, also big R, small r. And same thing for the last part right here. If you have the big R and the small r together, it forms big R, small r. So the F1 generation will be 100% big R, small r. Is that round or is that wrinkle?
Round.
That is round. Okay, so here's an important thing to remember. If both parents are pure, okay, the dominant trait always show up in F1. Show up in 100% of F1. If you have two parents that are different, two different traits, one dominant, one recessive, and then you breed them, 100% of the first generation will have the dominant trait. Okay. So what if we take the F1 individuals and then we cross them again? So we cross them again. So now follow to F2, right? So these are F1 individuals. And we knew that they were big R, small r. They all look the same. They all have the same genetic makeup. So when this plant makes gamete, makes the pollen, again, the chromosome separates. Half of the gamete is going to have the big R, and the other half is going to have the small r. So this one can go here. It doesn't really matter which one goes where. It's the same. So here, when the chromosome separates, big R and small r. So I'm going to give you a moment to fill in the chart yourself. What happens if this combines with this? What are you going to get? And so on and so forth. Just do all four combinations and see what you're going to get. Yeah, that's right, Mary. Okay, so if the big R here and the big R come together, you get that. This and this together, we get big R, small r. This and this together, also big R, small r. And finally, small r, right? So 1 out of 4 boxes is big R, big R. So that's 25%. Big R, small r, 2 out of the 4 boxes. That's 50%. And finally, small r is also 25%. So this is the genotype. But the phenotype percentage is different. What is the shape of the big R, big R? Is it round or wrinkle?
Round.
Round. Big R, small r is
Round.
also round. Right? So together, it would be 75% round. Small r is wrinkle, only 25%. Does anybody have any question about this? It's not too difficult, I hope. Okay, let's do some more practice. If a red flower is dominant over white flower for a certain type of orchid, write the genotype for the following flowers. So we have to come up with our own letters. We use the dominant trait to pick the letter. Dominant is red, so we'll say big R is red. And then we have to use the same letter, okay? You cannot use R for red and W for white, okay? We pick big R because red is dominant, and automatically small r is the other color, which is white. Okay. So homozygous red flower. What is homozygous red flower? Anybody?
Capital R, 2 of them.
Yeah, big R, right? It's red and it's the same. Homozygous means the same. Homozygous white flower.
Small.
Yeah. White is small r and it's homozygous. That means they have to be the same. Anyone else? Someone else for this one? Heterozygous red flower. Someone who hasn't been spoken yet in the class? Anyone? Or you can type it out if you want. What is heterozygous red flower?
It's capital R, 1 and small r.
That's right, big R, small r. Yeah, very good. What about heterozygous white flower? Anybody?
No.
No what? Anyone? What is this? What is heterozygous white flower? Okay, so this is not possible, right? Not possible to be heterozygous for a recessive trait. Okay? If it's recessive, it must be little r. It must be homozygous. Okay? So these two things contradict, not possible. Okay, let's try another question here. Consider a type of orchid where red flower allele R is dominant over the white. So same thing. Predict the phenotype and genotype of F1 generation when a hybrid red is crossed with a white flower. Hybrid red is crossed with a white flower. So hybrid red is big R, small r. Hybrid means different, okay? Cross means to breed with a white flower. Now the question does not tell you whether the white is pure or hybrid, but it doesn't matter because white is recessive. The only way to be recessive, as I just told you, is little r, little r. So we can go ahead and set up this chart. This chart is called a Punnett square. Punnett square. It's just, helps you keep track of the gametes, okay? So put it here. The big R and the small r will separate. And over here, it's just small r and small r. So if this combines with this one, we're going to have big R, small r. If this combines with this one, also big R, small r. This and this gives you small r, and small r once again. So in terms of phenotype, 2 of the 4 are going to be red, and 2 of the 4 are going to be white. And in terms of the genotype, big R, small r, 50%. Small r, 50%. Questions? Is everybody okay? You guys are uncharacteristically quiet after the break. Is there anything I can help clarify the situation for you, or we are all good? All right, no news is good news. Last one here. Why don't you give it a try? Okay, give this a try. Feel free to send me answers if you have, or you can wait. Okay, yeah, I'm getting some answers in. They look correct. Yep, another answer. Good. Anyone else want to share their answer with me? Okay, well, let's take it up. Pure red. Red is still dominant. So pure red is big R, crossed with a white. White is recessive, so by definition, it has to be pure. If you do the Punnett square, you will have big R, and small r, small r. And again, it doesn't matter where you put this, like you could do the small r on the side. You would have the same answer. And when you fill these in, you will see that it is 100% big R, which is 100% red. That's it. Okay. Now, the study guide has a lot of questions in it. You should work through them. And I'll post the answer on eCentennial. The answer is at the back anyways, okay? But like, you know, I'll post a solution, the full written solutions, so you can see it properly. Like I said, no synchronous class on Wednesday. But you know, I'll post a link. Actually, I'll post a link right now in the chat box here. Like if you're not tired and you have time, you can directly watch the link after class, right? Because that's going to be a continuation. Now, I reorganized the slide a little bit compared to what's in the video, but the informations are the exact same. Okay, so like you might see that the slide don't exactly match up with what the video is showing in terms of the order, but don't worry about it. Okay, all the information that you need to learn are in the video. Okay, so anything else before I go? Next Monday we'll have class next Monday. Okay, which is April 1st. Do we have anything on April 1st? Actually, no, right? I mean, no assessment. I don't think there's any assessment. No, nothing. I just confused you with my other class. So yeah.
I'm confused. So for Wednesday we don't have class?
We don't have face-to-face class, okay? Like I can't make it to live lecture, okay? So you will watch the recording instead.
Okay,
Is that
thank you.
yeah, all right. But so there are a lot of these questions, okay? You guys have to do it. I mean, you haven't learned these yet, okay? I only show you mono hybrid cross today, so you can do those, okay? But then there are dihybrid crosses. There's like incomplete dominance and sex-linked, and everything is in the recording. Okay? It's probably better that you watch the recording than the live session, anyways, because you can watch it at your own pace and you can rewatch it until you get it. But here is what I need you to do, okay? I need you to finish or attempt to finish as many of these questions as possible. And then at the beginning of Monday's class, I will take up some of these questions, okay? You can email me the questions you want me to take up. I'll do those, okay? If I don't receive any requests, I will just move on to lecture 9. All right, so the quiz number 4 is going to be happening. Let me just double check that so I give you the right information. Quiz number 4, give me a second. This is for online. Quiz number 4 is going to be next Monday. Okay, so still have quite a bit of time, April 8th. And like I said, is going to be lecture 8 and 9. But more importantly, you have 2 attempts. Okay, so that way, you know, you can do the best that you can. And we're almost done. Okay, we're almost at the end of the course. Some of you got your microscope lab back. I am still working my way through it. Hopefully, I'll have them all up by the end of tomorrow. That's it. Okay, thank you. Have a good week. I'll see you next Monday.
Thank you.
Thank you.
Bye-bye.